A very good morning, student. I am Anshul Sharma, your biology teacher, and in this video tutorial we will discuss about one of the type of cell that is known as prokaryotic cell. Actually, the term prokaryotic cell, the prokaryotic is made up of two words. So here you can see the prokaryotic word is made up of two words. The first is the pro and the second is the karyon. The pro word means primitive or first and karyons means nucleus. That means the cells with a primitive nucleus with a old nucleus, or you can say loosely organized nucleus, are called prokaryotic cell. If we go definition wise, a cell with a nucleus that lack a nuclear membrane, that kind of cell is known as prokaryotic cell, and examples of these cells are the all bacterias. So with the help of diagram of a typical bacterial cell, we will try to understand further features of the prokaryotic cell. So as we discussed, the prokaryotic cells are the cells that lack a membrane bound nuclei. That means the boundaries of the nucleus are well defined because nuclear membrane is absent, and these cells also lack all membrane bound cell organelles like mitochondria, like endoplasmic reticulums, Golgi body, chloroplast, and etc. They are also absent in a prokaryotic cell. So let's discuss the structure of a prokaryotic cell. The prokaryotic cell is bounded outside by a cell membrane, a plasma membrane that is made up of lipids and proteins, and this plasma membrane is further guarded by a thick cell wall. In the case of prokaryotic cells, the cell wall is not made up of cellulose. In the case of plant cells, the cell wall is made up of cellulose, but in the case of prokaryotic cell, the cell wall is made up of a substance that is called peptidoglycan. Inside the plasma membrane, the cell is filled with a fluid matrix, and this fluid matrix is known as cytoplasm. As you know, we are discussing about the structure of a prokaryotic cell, and in this prokaryotic cell the name is indicating the membrane bound nuclei is absent, and that is why the all DNA material of this cell is present in a certain region of the cytoplasm, and they have a circular DNA molecule. In the diagram you can see the DNA molecule is circular, and this circular DNA molecule is present in a specific region of the cytoplasm, and that specific region of the cytoplasm that contain the DNA. So this darkly stained region of the cytoplasm that contain the DNA, you can see there is no nuclear membrane that is separating the DNA from the cytoplasm, but there is a specific region of cytoplasm that contain the DNA, and this region of the cytoplasm is known as nucleoid. So prokaryotic cells don't have nucleus, but they have a certain region of cytoplasm that contain the DNA, that is called nucleoid. However, the prokaryotic cell lack membrane bound cell organelles, but one cell organelle that is not membrane bound. They are made up of proteins and RNA. The ribosomes are present in the cytoplasm of the bacterial cell or the prokaryotic cell, but there is a difference in the ribosome of prokaryotic cell and eukaryotic cell. In the case of prokaryotic cell, the ribosomes that are present, they are of 70S type. The meaning of 70S, the ribosomes are of two types, 70S and 80S. In prokaryotic cell, 70S ribosomes are present, and in eukaryotic cell, 80S ribosomes are present. S is representing the sedimentation coefficient. When we perform the centrifugation, so the ribosomes that settles down at 70 rpm, they are 70S, and that settle down on 80 rpm, they are the 80S ribosomes. So prokaryotes are having 70S type of ribosomes that are freely distributed in the cytoplasm. Except the ribosomes, the cytoplasm also have very small circular DNA molecules, and these circular DNA molecules are not the part of bacterial chromosome. This circular large DNA molecule is the bacterial chromosome. Single bacterial chromosome is there, but except this bacterial chromosome, some or other small circular DNA molecules are also present, and they are known as plasmids. We will discuss about the plasmids in higher classes, but they are also present in the bacterial cytoplasm. Some of the bacterial cells are motile, that means they can locomote, and for the locomotion the bacterial cells have a cell organelle that is called flagella. This flagella show viper-like movement, and with the help of viper-like movement of the flagella, the bacterial cell can move from one place to another place. But there is a difference between the flagella of prokaryotic cell and eukaryotic cell. If I cut a section of flagella and I look at the section of the flagella, it is only made up of single strand. But if I cut the section of flagella of a eukaryotic cell, the section will show the arrangement of tubules, 9 tubules in periphery and 2 central tubules are there. So 9 plus 2 arrangement of microtubules is there in the flagella of eukaryotic cell, but that kind of arrangement is absent in the flagella of prokaryotic cells. Except this, there are some extension, or you can say the evagination of the plasma membranes are also present in bacterial cells. Like the larger evaginations are known as the fimbres, and alternatively the smaller evaginations are also known as they are known as piles. So they are the fimbres and they are the piles. The fimbres are responsible for the adherence of the bacterial cell to any surface. If the bacteria wants to adhere to any surface, they will adhere with the help of fimbres, and piles are responsible for the transfer of material between the bacterial cells. Some other features of the prokaryotic cell. The prokaryotic cells have the ribosomes that are of 70s type. They have a single chromosome in the form of a circular DNA molecule, and this DNA molecule is naked. In the diagram you can see there is no protein that is associated with the DNA. In the case of eukaryotic cell, the DNA is a linear molecule and it is found associated with the proteins. So DNA is covered with the proteins in the case of eukaryotic cell, but in the case of prokaryotic cell, the DNA is circular and it is naked. The prokaryotic cells reproduce or produce new daughter cells only by the binary fission, or sometimes they show the fragmentation and budding, but they never show the normal procedure of cell division, that is mitosis or meiosis. So this is all about the prokaryotic cell and its structure. Thank you very much for listening and watching the video.